Right. Thank you very much, everybody. It's a bit of a strange experience because I'm talking to the audience who are watching online as well. Hi, everybody. Thank you for joining us. So this screen will now be being shared with you. So they're just going to see like it's Teams meeting. Yeah. They're actually not going to see us now.
They can hear me.
They can hear you.
Yeah.
I hope. There will be a chat running on Teams. So anybody who is watching remotely, if you've got any questions or points to raise, if you type into the chat, I'll keep an eye on that as well. So thank you very much, everybody, for coming. A short bit of housekeeping just to mention. Toilets are back out through there. I don't know if we've got anybody who isn't a student or staff, actually. I think everybody knows where we are. But they are through there. You carry on straight through and turn right through the double doors. There's no fire alarms, touch wood, but should there any go off, that's the fire exit there, so it couldn't be more straightforward. Professor Pete Nellist has very kindly come up from Oxford University as part of the Northwest Science Network, of which we are now a hub in the area, to talk about the work that he does in material sciences and hopefully inspire students to aspire to going on to study in this sort of area that he works in. So without further ado, I'll hand over to you.
Thank you, David. It's great to be here. I've had a really enjoyable day. It's great to be giving my first lecture here at Blackburn College. What I thought I'd do to start with actually is just talk a little bit about why. Oh, first of all, for those of you who weren't on the previous talk, I'll introduce myself. So my name's Pete Nellist. I'm a professor of materials science at the University of Oxford. So I lead a research group that uses electron microscopes to look at atoms and how atoms are arranged in materials and how they bond in materials to try and make the relationship between material structure and properties. If we can understand the relationship between material structure and properties, we can then think about how to make better materials for improved performance. And that's something that I'll be talking a little bit about in this lecture. And I'm also what we call tutorial fellow at Corpus Christi College in Oxford. So I suppose the first reason is I'm very busy. I've got a research group to run. I've got a lot of teaching. Why have I come up to Blackburn to give this lecture? Why am I leading this activity known as the Northwest Science Network? So if you just go onto the next slide, please, David. The answer is actually very simple. If you think about a lot of the challenges that are facing us now as a civilization, as a society, you can really start to realize that a lot of the solutions are going to be about science. We already saw when we got hit by COVID, the importance of science in both understanding how the virus is spreading and also in developing a vaccine, how important science was there. But also there's other areas, for example, when we think about climate change and energy sources that are delivering oxygen, carbon dioxide into the atmosphere, then obviously the challenge around low carbon energy is very much a science challenge. And in my own area, for example, we can understand that a lot of renewable energy sources, such as wind or tide or solar, for example, are very intermittent. The wind doesn't always blow, the sun doesn't always shine. And so energy storage is a very big area of research. And of course, when you start thinking about cars, automotive-based electric cars, then batteries, which is a form of energy storage, is an important area. And one of the areas that I work on is lithium-ion batteries. And actually, if you look at these batteries, which are now used in, well, you've got more in your phones and laptops and so on, but also now in cars, you can see how the red line here is actually a measure of the capacity of lithium-ion batteries over time from 1990 through 2010 up to the present day. And we can also see how the cost of lithium-ion batteries has come down. So there have been huge advances in batteries, but we need to go further. We need to have better ones, so the cars have a longer range. We don't need to keep stopping to recharge the cars and so on. So a lot of the work we're doing is on batteries. So these are huge challenges, and basically, if we're going to be able to address these challenges, we need to encourage and inspire your generation to actually come with us and do world-leading research to be able to solve some of these problems. So I basically want to get out to as many places as I can and encourage your generation. Younger people are the most creative and actually do the really creative science. And so I want to encourage your generation to come and join us. So if you just hit spacebar there, David, actually help you spacebar. Yeah, will it work? Yes, there it does work. we have a need for scientists. So that's you lot to come and join us. And so that's really why I'm doing this. Let me just talk about what how the Northwest Science Network works. Basically, what we're aiming to do, first of all, is to enhance subject knowledge by having lectures such as this, where we come in and talk about what the frontiers of research is. But we also have other activities where we get to interact a lot and raise ability to explore and communicate interest in science, various events that do that. Keep going, David, thanks. Yeah, raising the profile of non-A-level subjects to really have people thinking about what they might like to progress to at university, and encouraging thoughtful course selection. And really to raise aspiration and ambition as well, where if you decide you want to study a particular subject, where are really good places to go to study it, and not be put off by weird and wonderful myths that are usually a load of rubbish, but actually to actually just go to somewhere we think would be really good to do it. And also to work with schools and communities in the study of science. So that's what we do. How it works, we lead it from my college in Oxford, Corpus Christi College. We work with 3 hub schools, Cheshire College South and West, various sixth form in Manchester, and now Blackburn here. They provide the regional and logistical support for events and interact with schools and colleges in their region. And then we coordinate things like the academic resources and the leadership, working with partner HE institutions. So that's essentially how it works. And these are the sorts of activities. We have a launch event, which was actually something you may have attended, was down in Crewe in October. Twilight lectures, which is what this is. University interview workshops. We talked a little bit about that in just about half an hour ago. We arranged laboratory visits, an Easter maths workshop. If you keep going, David. And then a summer school, which is great fun, down in Oxford, where you can come down and join us and do all sorts of activities, poster projects, small group tutorials, lectures, laboratory sessions, people from lots of different schools all mixing together. That's in June. It's a lot of fun. Everybody always has a really fun time in it. So that's something to look out for. And this is targeted at year 12. So the summer school is really at the end of year 12, and the program is really aimed at year 12. But we do have quite a lot of year 13s want to come along with things, and that's fine. And sometimes year 11s as well. And that's also fine for many of the activities. And there's a website which you can, or just Google Northwest Science Network, and you'll find the activities. So that was a very whistle-top tour. I didn't want to spend too long on that. Let me now move on to the science and actually start talking about what I do. So my title is Seeing is Believing: How Observing Atoms in the Electron Microscope Helps Further Physics, Chemistry, and Biology. So that's really what we're going to talk about. So just click on to the next one. So I'm a material science. Put your hands up if you've ever heard of material science. This is really one for the students. That's not bad, actually. Oh, some of the teaching staff over there. That's a relief. So it's not an A-level subject. It's one of these subjects which you don't necessarily hear about being a separate science when you're saying an A-level student, but at research level, it's massive. There are absolutely enormous research conferences that I go to. And the reason is that if you think about it, what material science is around the development of new materials, or what we use every day. You can look around a lecture theatre like this and you see metals on that metal stand. There's polymer, there's plastics on the surfaces here. There's fabrics. These are all, you've got a plastic bottle. These are all materials, and this is what we actually think about: is how do we make better materials? How do we do that? And of course, if you think about it, that's hugely important to industry, and so it's a really well-funded and a huge research area. And this slide, which is a bit of an old one now, sort of illustrates the sort of evolution of material science, starting right back in prehistory, starting back in 10,000 BCE here. Historians know how important material science is because they've named whole historical eras after materials. Stone Age, Bronze Age, there you go, you're a historian, yeah. Iron Age, yeah. Materials, right? So historians are really material scientists, they just don't know. Just for you. As time went on, obviously, you can see here in 10,000 BCE, we got things like stone, straw, and brick, and wood and skins, and so on. And then the metals started to appear. We've got the Industrial Revolution in the 19th century, where we started to see steels come now, so use of steel. And then as time went on, particularly as we moved into the 20th century, the development of aviation, more complex alloys, which are mixtures of metals that can give us enhanced properties, lighter alloys with enhanced strengths and so on. But also then the emergence of nylon and polymers and plastics and things like that really in the 20th century as well, and much more complicated alloys as well. You'll notice there's a real change in the sort of post-war period of the material science. And most of the university departments, material science university departments, emerged in the post-war era when the importance of materials as a separate science discipline really started to emerge. But there was also something else that happened in the immediate post-war era, which really helped open up the field. And so if David can just click on that, and that was the development of this thing. This is one of the very first commercial electron microscopes ever delivered in the UK. It was made by a German company, Siemens. It's a Siemens Elvis Cop 1, and it was in the lab of this guy, Peter Hirsch, who was actually head of my department for 24 years. He retired in 1997, I think, but he's 97 now, still going strong. We still have conversations with him. Amazing guy, doing really well. And it just shows you that being a material scientist is good for your health. I don't think that's in our prospectus actually. No. So cause and effect. Yeah, absolutely. But we'll talk a little bit about this microscope in a minute. It was actually bought primarily to look at biological materials. Actually, they realised that you could actually see things, subcellular structures, and so it was really. But he had other ideas of what he wanted to do with it. So we'll come back to the biological applications a bit later in the talk. But if we click on, just to give you a quick ray diagram. Hands up, students, who's used a microscope? Hey, excellent. Most of you have got your hands on a microscope. And what was the radiation that we used in it? Are you confused now? Was it light? Was it light? You still used a light microscope, indeed. And most microscopes essentially work in a sort of similar way, in a way. What we've got, this is actually a diagram of an electron microscope, but we've got a beam of electrons coming down, or it could be light. Here we've got a thin specimen, which the radiation is going through, and it scatters as it goes through the sample. And we have a series of lenses that just magnify it, and we form an image at the bottom. And that's essentially the layout. So if you just click on David. So what was he actually interested in looking at? And one thing I wanted to point out to you as well, because I'm going to show you some movies in a minute if they work, is this is in the 1950s. There's an old-fashioned movie camera, 16mm movie camera with film. You don't know what film is, I know. Looking down through the window of this thing at where the image is formed on a so-called phosphor screen. And that was how they just sort of mounted this camera to try and take some movies. And I'll show you these movies in a minute. Well, what was Peter interested in? If you just click on to the next slide, David. Yeah. I'm going to show you what I think is the most incredible experiment in materials science. Right, are you ready for this? Here's a paperclip. I'm going to take the paperclip with my finger and I'm going to bend it straight out. Isn't that amazing? Now, no, what you're not looking too impressed. It's like, oh yeah, the bloke from Oxford came up and he bent a paperclip. I thought it was really clever. Why is this a really amazing thing? The question is, is it bent so easy? Why was I able to bend it with my thumb? And why didn't it break when I tried to bend it? Because almost all materials are crystalline. So the atoms are arranged in a completely regular periodic lattice. This is actually sodium chloride. It alternates sodium chlorine in a completely regular way. So when you start to think about trying to bend a crystalline, this is a steel, but it's got iron, and it's going to have a lot of this will be crystalline as well. A lot of crystals in it. When you think about bending something that's crystalline, you immediately run into a bit of a problem. So if we click on, here we go. So here's a crystal, just a sort of schematic crystal. When we're thinking of something bending, what we're really talking about in terms of the atomic arrangement of atoms in a crystal is that the atoms actually need to slide over each other. We call it a shearing process, right? The atoms have to shear. We need the sliding of the atoms past each other, and that will allow movement within the material, which allows the material to bend. And there's a bit of a problem when you think about a shearing process in a crystal. Now, David, if you put your mouse somewhere around there, there should be, yeah, there we go.
Oh, excellent.
Movie thing, let's see if this works. There we go. So this is the shearing process going on. I'm sorry, David, I'm being really boring, but if you can keep clicking on it and keep repeating it while I was speaking, that would be really great. You can see there's a few problems here. Oh, no, hang on. No, go back. There we go. There we are. You can see that we've got all these bonds, atomic bonds between these atoms. And as we shear it, they essentially all break at the same time. So what that basically means is, as you sheared it, the material should just fall apart. It should just fracture. And so, but it didn't. It just bent. It didn't fracture. Also, with the theoreticians, people like doing calculations on things, calculated the force involved with distorting and breaking all these bonds. It was very, very high. This is much softer, way, way softer, much easier to bend with my thumb than any of the theories could predict in terms of this shearing, this block shearing model. So this was a real problem. This was, even into the 20th century, nobody could really explain why metals bent so easily. There wasn't a model for that. So one suggestion, if you click onto the next slide, David, was that actually there may be crystalline defects responsible. And we, as material scientists, we spend a lot of time talking about defects. And what we mean by that is where the regular arrangement of the atoms in the material has broken down in some way. You can see there's an extra plane here of atoms in this crystal that's come to a halt right there and stops, and the other planes kind of close up to accommodate the gap. We call this a crystal defect. One of my colleagues, Holly Humphreys at Queen Mary in London, says, you know what, Pete, crystals are like people. It's their defects that make them interesting, which I think is called quite a good turn of phrase, actually. And so we spent a lot of time looking at defects. And this dislocation had been, this is called a dislocation, where this extra half plane runs out. This was a sort of theoretical construct, really. People said, well, this might explain why metals can bend so easily, why they're so ductile as we use it. So we've got to the next slide, David. This is the model that really applies. If you think about having a dislocation, it allows you to move this dislocation, this extra half plane, through the crystal, breaking one row of bonds at a time, not breaking the whole plane of bonds, but only breaking one row. When I say one row, we're looking through the crystal in projection this way, so we're looking right through it. So each of these blue blobs is a row of atoms that we're looking along, and we're breaking one row of atoms at a time as this dislocation rolls through. And that was potentially an explanation of why metals could bend so easily. The problem was there was absolutely no experimental evidence for this whatsoever. Nobody had actually ever seen a dislocation. This was back in just about the pre-war period and the post-war period, about the 1940s and 50s. And they were hugely controversial. The theoreticians were just having a great time doing all sorts of calculations of how dislocations can explain why metals are relatively bendable. And everybody else going, well, hold on, we've never actually seen them. We don't know if these things exist. So that was what Peter Hirsch was interested in. So if you click on the next slide. So he decided, and I've still got the letters he wrote, he got some people, some suppliers of metals to beat them with literally, if you ever come across gold leaf, it's actually made by beating gold. And he made very, very thin layers of gold and steels and aluminium and put them in the microscope, in this microscope. He sort of persuaded the biologists who owned this, he could put a bit of metal in and have a look. And I'm going to show you what he saw when he did his movies. Here we go. It's a bit, so if you click, actually, David, I think I'm going to ride on this one if I may, because that's what I know when we'll start it off. Now I know this looks like, you know, something. This looks like one of these silent movies, doesn't it? You almost expect to have some sort of piano playing alongside it or something. We don't need all the titles. I'm just going to kind of just shift along pretty quickly. So this isn't steel, actually. This is basically what he saw. I know it looks a bit blurry. Ignore these blobs here. These are just marks on the screen. But what he saw in this electron microscope were these lines that were then moving as the material bent. He actually heated up inside the microscope, and these lines started moving. And this was the first basically it's just a screen with a lot of parallel slits in it. And so when you have a screen with a lot of parallel slits in it, you get a process called diffraction. Let me illustrate what diffraction is actually. So this is a quite powerful laser pointer, which I don't use very often. I point at the ceiling, there we go. But if I was to put a so what these are that I can screw onto the end of them is actually diffraction gratings. They're just a set of regular lines in various arrangements. And if I do it, you can see, ooh, it spreads the light out, and it sort of spreads out and creates these amazing patterns, which you can then see up there on the ceiling. So that's how diffraction, what diffraction is. It's a spreading out of the rate of the radiation, and that's what is essentially happening as the radiation you're using in the microscope is passing through a sample. So you get this diffraction process which spreads out the light, and then the job of the lens is to do what we call refraction. Brings the rays back, and they all interfere and overlap with each other, and eventually form an image down here, where is what we call the image plane. Now, the key thing about diffraction is as the slits in this grating get closer together, the angles of diffraction get larger. These angles of these beams get wider. So as the features get smaller in your sample, it scatters out to larger angles. And what that means is you need a bigger lens to collect that scattering and then to form the image. If the lens isn't big enough to collect the scattering, you can't see those features that are that small distance apart from each other. We call this the resolution of the microscope. And there's a more fundamental limit as well, that if the spacing between the features that you're trying to image in the microscope, if they get smaller than the wavelength of the radiation that you use, then no diffraction occurs. Actually, it doesn't diffract anymore. And so no matter how big your lens is, you're not actually going to collect any information about those small features. So what Abbe realized is that you can never see things in a microscope that are smaller than the wavelength of the radiation that you're using to look at the sample. So here's a quick question again for the business. Perhaps anybody know what typical wavelengths of light are? Ooh. You're going, oh, yes, gotta give me a guess. It doesn't matter if you're wrong. 100 units. Pretty small. What's that? 100 microns. You're sort of heading in the right unit space. You're about 100 times too big, actually. It's about 400 to 700 nanometers. So about half a micron is a typical wavelength of visible light. So that's about the smallest thing. A micron is 10 to the minus 6, a millionth of a meter. So you're kind of limited to that sort of size. That's the smallest sort of size of things that you can see in a light microscope. And there's an awful lot of stuff in all sorts of areas of science, biology, chemistry, and physics that are smaller than that. Atoms, the spacing between atoms in a material are about 1,000 times smaller than that, the distance between atoms. And so you haven't got a hope of ever seeing an atom in a light microscope because they're too close together, and the radiation of the wavelength, the wavelength of the radiation, I should say, is too long. So that's a fundamental problem. In hopeless with light, you're not going to ever be able to see an atom. So that's why, if we click on, there's why we do it with electrons. So one of the sort of weird parts of 20th century physics is something called wave particle duality, and that's the electrons, which we think of as subatomic particles, charged subatomic particles. Actually also have a wave nature. They behave as waves. And this chap, Louis de Broglie, had a formula to calculate what the wavelength of the electrons are. And I don't want to get into the details of how it's calculated. But in my lab, we have electron microscopes. Actually, the one that we use mostly is actually powered by a 200,000 volt power supply. So that's about 1,000 times higher than what comes out the mains of the socket. So it still sits in a lab, but it's a pretty big thing. Have the power supply for that. You don't want to put your fingers in it. It'd be a bad idea. If you accelerate electrons through that potential, 100 or 200,000 volts, you end up with electrons that are traveling just over half the speed of light. Actually, they're zipping on pretty quickly. And if you then use de Broglie's formula to calculate what their wavelength is, it's 100 kilovolts is 3.7 picometers. So 200 kilovolts is actually 2.5 picometers. A picometer is 10 to the minus 12, so that's a millionth of a millionth of a meter. This is about a few hundred times smaller than the distance between atoms. So the wavelength of electrons should in principle be perfect for being able to look at atoms. And so that's why we use electron microscopes, because it takes us beyond the resolution of light. Light takes us to about half a micron. In principle, electrons can take us right down to the atomic scale, because we have such a short wavelength of electrons. So yeah, as I said, just click forward. So how do you focus though? I mean, there's a few things we've got to do. It's not going to be like we can't put a glass lens in now. If we have a glass lens, the electrons will just slam into the glass and just stop and not go anywhere. So we can't use glass as a lens. What we actually use is magnetic fields. We have big copper windings that we wind around the beam line of the tube that the electrons travel down. And we have things like iron here to direct the magnetic field and generate a very, very intense magnetic field, which we can use to focus the electrons and make electron lenses. Also, the whole thing has to be in vacuum as well, because the electrons got to be in travel. So it's actually got to be a big pumping system to make the whole thing in vacuum. So, and these electron lenses have very high currents in them. When I say very high, sort of 10 to 15 amps or so. So they're getting quite hot. There's also water cooling pipes as well to cool them down, and all sorts of stuff. So they're quite complicated devices. We've just ordered a new microscope in my lab actually, and the price tag is funded by the taxpayer. So I'm slightly nervous of telling you, those taxpayers here, how much it was, but it was 3.25 million. So yeah, that's the sort of typical price tag for a state-of-the-art microscope these days, about 3.25 million. Now, I mean, this is all, as you saw, these microscopes started becoming available in the 1950s or so. But I mentioned a few minutes back that we only started seeing atoms really properly in the last sort of 15, 20 years or so. So what's the problem? The problem is that these lenses turned out to be rubbish. It's against the laws of physics to be able to make a good lens shape out of a magnetic field. Magnetic fields have to obey certain laws called Maxwell's equations about the shapes they can have. And it turns out you can't make a magnetic field into a very, very good lens shape. It's actually a really rubbish lens shape. And it's a bit like making a light microscope or something, or a magnifying glass out of maybe a wine glass or something like that. So if you imagine, trying to magnify, if you sort of look through that, you can see it's got a lot of distortions in it. And if you think of, if you've got a glass this shape at home and put some water into it, you can see it'll just you try and use it as magnifying glass. It's really rubbish. It doesn't really magnify well. It's got a lot of distortions. We call it spherical aberration. It's so we ended up with these lenses which were absolutely rubbish. In fact, if you do the calculation, if our eyesight had the same quality as an electron lens, we would all be legally blind. So you spend £3.25 million on a microscope, which is essentially legally blind. I mean, this is kind of a real sort of this would be one of the real tensions in microscopy for a long time. But if you click on the next slide, about 20 years ago, we found a solution, and actually the solution turned out to be technology which we sort of we learnt of from particle accelerators. Have you ever heard of CERN and some of these particle accelerators and so on? So they use quite complex optics, which are non-round. Things called octopoles, which have these apoles and quadrupoles and things like that, which can squeeze beams into weird and wonderful shapes, which is what you need to do when you're doing particle optics at CERN or in a synchrotron or something like that. And what we learned was that, well, actually the principle goes back into the 1940s, but we sort of learned that by combining lots of these non-round devices together in a stack, you could actually make what is essentially a pair of glasses for your microscope. It actually fixes the bad vision. It makes something that has exactly negative aberrations that the opposite aberrations that the main imaging lens have. So it fixes it in the same way that glasses fix your vision. But we needed to put several of these non-round optics together. So as the beam passes through the optics, they get squeezed into all sorts of shapes and then comes back out again. And actually, one of the great things about getting a good science degree is it can often open up all sorts of different opportunities. And in 2000, when this technology was emerging, some people I've met when I worked in the US for a bit started to set up a company to actually make these devices in Seattle. And they kind of emailed me and said, did I know anybody who would be interested in joining the company? And I said, yeah, me. I'd like to come and do that. So I left. At that time, I had a lecturing job at the University of Birmingham. I left it. I went up to the US with my wife and worked with this company making these spectacles for microscopes. So if you click on the next slide, these two gentlemen, Andre and Nicholas, who are my colleagues, this was our Mark II spectacles. It's about 14 centimeters long. I know it's gold coated. It's not solid gold. It's gold coated because it stops it corroding some of the optics in there. It's got 7 layers. These are the non-round optics, and we could fit these to existing electron microscopes and essentially put these in the spectacles and make the vision better. Now, a bit more expensive than spectacles. If you wanted to buy these, about £500,000. So each one about £500,000. Cheaper than a microscope. So, you know, that's not to like. But yeah, and the company's actually, I decided to come back to the UK and become an academic again, actually. But the company's still doing well. I still see Andre and Nicholas regularly, and we keep in touch, and things are going well. So that was really the key, actually, was we can actually now have glasses for the microscopes, and that allows us to see atoms. And this is what atoms can look like when you're looking at one of these microscopes. This is actually the bottom here is actually manganese Mn3O4 for the chemist. This is actually a material which potentially could be used in something called a supercapacitor, which is a bit like a battery, but can deliver electric power very, very quickly. If you need a real surge of electric power, these supercapacitors can deliver this power very, very quickly. And the Mn3O4 was a candidate electrolyte for the supercapacitor. And you can see the Mn3O4 is quite a complicated structure. What you're primarily seeing here, the manganese atoms, in this particular mode of imaging, it's a little bit hard to see the oxygens. These are manganese. Where there are these brighter blobs, this is a crystal we're looking all the way through. So it's looking in projection all the way through a crystal. Where they're brighter, we've just got more atoms. The atoms are like a higher in density at that point, looking through the crystal than they are here, which is why they look a little bit brighter at this point. This is quite a complicated crystalline structure that's known as a spinel. Now, what we found actually when we started looking at this material is it's actually quite sensitive to the electron beam, and that's one of the challenges with electron microscopy, is that the materials can be damaged and changed because actually the radiation level, when you've got this high intensity electron beam hitting samples, the radiation level is very high. You wouldn't survive if you were sitting in it with all these electrons hitting you. And so often you find materials can actually start to be modified by the electron beam. That's something we always have to be very careful of. In this case, it started happening, but it actually started happening at a nice speed. And David, again, there should be a movie thing at the bottom of the there we go. Click on the arrow, and it should just run. What actually started happening is that this started changing as the electron beam from Mn3O4 into MnO. And MnO actually has the same structure as sodium chloride. It's called the rock salt structure, which is a much simpler crystallographic structure. And here it is right here. And as the movie runs, you can sort of see how the atoms, I think one of these pairs of atoms, maybe it's that one. If you can see that, they can't decide whether they want to be in the rock salt position or the spinel position. They sort of bounce backwards and forwards a few times before they finally go. And so we were able to watch what we call a phase transformation, a material change from one structure to another structure in real time in the microscope, which we thought was actually quite good fun. So it made a rather nice movie. So this is one of the things I like to show first, is how we can now see these atoms moving as it's changing from one's crystal structure to another crystal structure. So moving on, actually, yeah. So go back one day. Before we, I don't want to move on to this quite yet. So before we move on to our next slide, I want to ask you. This is going to do a little bit of chemistry now. Who's got some gallium nitride? Anybody got any gallium nitride? Oh, I think you have. I'm sure you have. Right. But you're all thinking, well, I've got some gallium nitride now. Anybody got, I don't know, LED bike lights, LED lighting at home? Yeah, that's where your gallium nitride is. So if you then click onto the next slide, yeah, low energy lighting. When I was your age, lighting was done by incandescent light bulbs. This is going to seem like the Stone Age to people, but it's the way it worked was that you had a tungsten filament wound in a coil, which you passed your electricity through, and the coil just basically got white hot, and that gave off light. But it gave off an awful lot of heat as well. That was basically the problem. So they were hugely inefficient. An incandescent light bulb about 5% efficient. Most of the energy you're putting into an incandescent light bulb, which is one of these where it gets hot, comes out as heat, which is not really what you want. And about lighting uses around almost 20% of the world's electricity production. So this is a serious issue, that actually all this lighting was just pumping out a lot of heat, which doesn't seem like a very good idea. So modern lighting is based on light emitting diodes. Now, again, when I was your age, light emitting diodes weren't that common. You could basically get them in red, green, or orange. They're the three colors you get light emitting diodes in. And if you want to make white light, that's a problem, because the one you're missing is blue. And you can't make white light unless you start mixing red, green, and blue. So you need blue in there in the mix as well. And there were no blue light emitting diodes. And the reason is that you need the way that light emitting diodes work is that the electrons inside the material, when you start passing current, move between what we call bands, and the electrons can sit in certain allowed energy bands inside materials. And as they move between these bands, they can give off light. And so what controls the wavelength of the light is the gap between these bands and how much the electrons change energy as they move through the material. We needed something that blue light is higher energy. So we needed a wider band gap, something with a bigger band gap to generate the light. And gallium nitride is a wide band gap semiconductor, as we call it. The problem is, you've got to make it. And you can't go to a gallium nitride mine and dig it up. You have to grow it. You actually create it in the lab. And you grow it on substrates such as sapphire or silicon carbide. These are expensive, but also the crystal structure of the gallium nitride doesn't match very well to these. So when you grow it, it actually has, guess what, a load of these dislocations in again. So similar things that Peter Hirsch was dealing with, but now in a semiconducting material. And these dislocations really reduce the efficiency of the gallium nitride. Where you have a dislocation, the electricity goes there. It generates heat rather than light, and you lose efficiency. So we're very interested in looking at dislocations. So if you just click on David. So here is a picture that we took in my lab of a dislocation in gallium nitride. Gallium nitride is a hexagonal material, so you can see that it's hexagons, the crystal structure. But there is a dislocation there. It's quite hard to spot. You might be able to see that there's extra planes of atoms coming in from the right-hand side that stop right in the middle, and then the planes of atoms either side shear across a little bit, swerve to close up the gap. A little bit hard to spot, but you might just be able to see there's the shearing across of the atoms and the extra half planes coming in from the right-hand side. What we're really interested in is what the atomic arrangement is right at where the extra planes run out in this crystal, right at the dislocation core. And so this is actually, we've now achieved, if you like, the dream that I talked about before, where we really want to look at the atomic arrangement at the core of dislocations. Now, here's an interesting thing. If you look at this dislocation, most of it's hexagons. Here at the dislocation, there's a 7-membered ring, as the atoms have formed a ring of 7 atoms. And there's next to it a ring of 5 atoms right there. So it's formed what we call a 7-5 arrangement. So rather than having hexagons, it's gone to a 7-membered ring and a 5-membered ring. This is because of this defect. This is the defect in the crystal where this extra half plane runs out. Now, thinking about gallium nitride, where it normally forms hexagons, and you've got gallium bonding to nitrogen, and nitrogen bonding to gallium, and gallium bonding to nitrogen, and so on. Can anybody think of a problem with either a 7-membered ring or a 5-membered ring? Any ideas? Just thinking about what I was saying with gallium bonding to nitrogen and nitrogen bonding to gallium. What happens in it? Yes. You either then have to end up with gallium bonding to gallium or nitrogen bonding to nitrogen. This is not good. Chemistry doesn't like this, right? It wants to be gallium bonding to nitrogen, nitrogen bonding to gallium. You don't want gallium next to another gallium or a nitrogen next to another nitrogen. This has got the technical term of a wrong bond. So you've basically got these wrong bonds in this structure, and this then is what starts to mess up the whole electronic arrangement in the material and causes the loss of efficiency. But then we went on and looked around a little bit more, and again, if you just click on, and found a much more complicated looking thing. Where there's an extra half plane in this one now. It's actually coming from the left-hand side, and the planes of atoms are closing up, going that way. And it's quite an elongated structure now. We don't have the 5-7 rings in it anymore. It's got this kind of long thing in it. And we actually looked at this for a very long time, trying to work out what on earth was going on. And the way we actually eventually solved it, I'm afraid, was going back to the good old-fashioned stick atoms together in a model, and actually make a model and see if we could actually explain it in terms of the model. We did, and this is the model we came up with. And if you click on the next slide, it's actually got a very technical term to it. It's a dissociated core structure with double kink. So that's what we discovered it was. I'm not going to explain what all that means, but this is the model right here. And you won't be able to see this online. I'm sorry, but you just have to trust me, and people can verify this, that actually the rings are now 4-membered rings, if you look down that now. So we've got 7-membered rings. And can you see the 4-membered rings looking through it in projection? That works again, doesn't it? If you've got 4-membered rings, you can have gallium bonding to nitrogen, nitrogen bonding to gallium. So this actually dislocation the core is we think, I mean, we need to do the modeling now, we think would be much less impactful on the performance of the material. So we can actually, if we can grow it really making these sorts of dislocations, we think we should be able to make better gallium nitride materials. Okay, time's clicking on. I've got quite a few applications. I think I'm going to skip over these, David. Can I have take over and jump past these? It's a little bit technical. Lots of maths, which I don't want to kind of terrify people with. I do want to talk about this. Who's heard of the hydrogen economy? That's it. So the idea here is that one of the fuel sources that might be useful is hydrogen. Because when you use hydrogen as an energy source, one of the ways you can use it is using something called a hydrogen fuel cell. And what the hydrogen fuel cell can do is generate electricity and heat. And the waste product is H2O. This tends not to be too much of a problem. It doesn't sort of pollute the planet like carbon dioxide does. There are a lot of challenges around hydrogen, using hydrogen as a fuel, not least transporting it and storing it, because hydrogen is such a small molecule, it tends to diffuse through a lot of things, including a lot of metals. So it just leaks out of pipes and things like that. So what we can't do is just pipe hydrogen down the existing gas network. The existing gas network is designed for methane, which is what natural gas is. And so we can't pipe hydrogen down it, sadly, because it will just diffuse out. But nonetheless, if you can solve these problems, these hydrogen fuel cells are very interesting. There is a challenge with the hydrogen fuel cells, and that's that at this point here on what we call the cathode side, we need to do something called the oxygen reduction reaction. We're going to have oxygen, O2 coming in, O2 gas, and it needs to form H2O. So we need to break the O2 molecule and split it apart. So we actually just have single oxygen molecules that we can then react with the hydrogen. That will use electricity will get involved. This is a form of science known as electrochemistry. Electrons get involved, and we actually have electron flow, and that's the electricity. So the thing that actually limits the efficiency with which these things work is actually the rate of this oxygen reduction reaction, splitting the oxygen molecule into these two. And to make it work, we have to use a catalyst. Now, A-level chemists, any here? Yeah, good. What's a catalyst? Speeds up the reaction. That's exactly what we need. It's not consumed by the reaction. And the one that works really well for the oxygen reduction reaction is platinum. Problem with platinum, really expensive. Platinum, so it's always typical, isn't it? The thing that works turns out to be really expensive. And actually, use of pure platinum as the catalyst here makes these things economically unviable. So, so one of the things that we've been interested in doing, if you click onto the next slide, is the idea of this is actually a research project. It's my former research student. He's just recently gone back to China, Shenan, and he came to Oxford, China, worked with me, did his PhD, which is a research degree. And what we were interested in doing there is understanding what happens if we mix platinum with something a lot cheaper, such as transition metals like nickel or cobalt or iron. We can use less platinum, hopefully, and maybe we'll still have a good catalyst. The original idea was that actually you have the cheaper metal in the middle. Actually, what I should have said is, sorry, maybe you just go back. The surfaces on which you actually do this catalysis are actually really, really tiny little balls of the metal. We call them nanoparticles. They're only about 3 to 5 nanometers in diameter. They've got about 5,000 atoms in them. They're very, very small because they've got lots of surface, basically. You make them very small, they've got lots of surface. And it's at the surface that the reaction goes on. So we make them very small. But what the idea was that we'd have in these nanoparticles, we'd try and make nanoparticles where the core, the inner side bit, was the cheaper metal, and we just had the platinum as the outer coating, which is where it sees the gas, and which is where the catalysis goes on, and that would make a cheaper catalyst. So that was the idea. So if you click on, but actually when this was tried, this is before we got involved. Actually, a number of groups did this. It turned out that actually these particles made up of platinum and other transition metals worked even better than platinum. This is a plot sort of essentially of activity. They work even better than the pure platinum. The problem with them is their stability. They don't last as long, but they work. But they're very they have a lot of activity. We don't really understand, to be quite honest, why. Why do they work better? If we can understand why they work better, we can then think about how to design catalysts that are better. So often in science, we find something works well. Why does it work well? We don't understand why it works well. What's going on? And so if you click onto the next slide, one of the things that people often think about, and it's coming back to these idea of bands, that the electrons can exist in bands. You may have heard of S and P orbitals? Yeah, have you come across those? What happens these are the levels in an individual atom. There's also D, which should be on here. Transition metals actually have a partially filled D band, the D orbital. When these atoms move close together, we have a lot of atoms close together, as in a metal. All of these various orbitals in the atom start to mix together, and the energy is spread out to form these allowed bands, allowed energy levels that electrons can exist in. They can exist in these levels here, but not in this so-called band gap right there. And we sort of got to the point where we understand that what makes the oxygen reduction reaction work better is that the D band, which is formed in the D orbital, is not shown on this diagram, but forms this D band, moves slightly in energy, and that actually just makes the catalysis work better. It affects how the catalysis works. But we don't understand why the energy of this D band shifts a little bit. So if you go on to the next slide. Yeah, so this is just to show you this periodic, hopefully you know what this is, periodic table. Basically, these are all the D band or the transition metals here. So we're dealing with platinum, and we're dealing with things like cobalt and nickel. We particularly looked at platinum, cobalt, which have all these partially filled D orbitals and a partially filled D band. And we were interested to see why does the D band move in the way it does. So again, so next slide. And some previous work by others has suggested that actually what happens is if you make what we call an alloy, which is a mixture of platinum and cobalt, the atoms sit closer together in that. Sorry, this is platinum copper, sorry. Copper is a smaller atom than platinum, essentially has a smaller atomic radius. And so that means the atoms sit closer together in the platinum copper, and it shrinks. And that actually then applies what we call a stress to the platinum and makes platinum get pushed a little bit closer together. When the platinum's in contact with the platinum copper alloy, which the atoms close together, it also compresses the platinum. And so measurements have been done using diffraction methods, which sort of showed that the distance between the atoms, even in the platinum shell, was smaller than you might expect for bulk platinum, for a big lump of platinum. So the idea is that some sort of compression, the platinum atoms being pushed together, is what controls the D band. So we had a look at this in our fancy electron microscope. So let me just click on to the next one. Very quickly, what we're able to do in our microscope, but I don't want to talk about this in detail. It's actually a big collaborative project with ourselves, University of Southampton, who've done the modelling, and an industrial company, Johnson Matthey, who are a catalyst company, and so a lot of this stuff. We can look at both what type of atoms are where. We can look where the platinum is, where the cobalt is, using spectroscopy methods, which I don't want to talk about in detail, but we can understand where different elements are in materials using spectroscopy in the electron microscope. And we can then also use images if you click on them, to look at where these atoms are. And there was an awful, I don't want to scare you with this, but there's, you wonder why maths is important. We need an awful lot of maths to be able to turn these images into measurements of how the atoms have moved. Lots of nice matrices going on here, trigonometry. Keep going, David. We've all got worse, all sorts of stuff going on. But we can calculate how the atoms have moved inside the material. And if you click again, David, what we basically realized actually is the picture's a lot more complicated, as it often is with these things. What we're basically showing here is what we call the strain, which is how the atoms have either been pulled further apart or pushed closer together. And it turns out that around the circumference of the particle, in its circumferential direction, you can see there's these little lines here which are blue. What this means is the atoms are being pushed together closer in this direction. But because they're being pushed closer together in this direction, you can see these other lines, which are red, means the atoms actually bulging out. They're being pushed out in the radial direction. So it's actually a much more complicated picture. It's not they're just all being pushed together. They're being pushed together in one direction and spreading out in another direction. If you imagine pushing something, it's going to spread out that way, and that's essentially what we're seeing. So now we're moving on to actually trying to understand how this movement of the atoms affects this D band. This is what's causing the enhanced activity, the fact that the atom's being squashed in this way. Right, at 6 o'clock, I should probably wrapping up. I do want to talk about some biology. If you just click me through, yes, right, we'll finish up. What's that?
Perfect time. Perfect stuff about some biology.
It's still about biology. What's that? It's what? Anybody know? It's a virus. Which one? Yeah, it is. I'm afraid I had to show it, didn't I? Yeah, there's the spike proteins on the side there. So you've probably seen lots of pictures like that in the media. That this is what COVID looks like. There's all these things are going to get you, and this is what it looks like. So you may wonder, how do we know it looks like that? What do we do to know that it looks like that? And that brings me on to the subject of structural biology, which is a huge area. And if you click forward, David, this is an interesting one. Look, it's kind of, you can see it got this oxy and then deoxy. It's a protein that can take up oxygen and then release it. Anybody know a protein that can take up oxygen and release it? Anybody know biology? Nobody biologists. No. What was that? No? It's hemoglobin. Yeah. So this is the hemoglobin molecule that's taking up oxygen. It's got different configurations when it's oxygenated and when it's not oxygenated. How do we know? How do we know it looks like that? How do we actually determine? These are called biological macromolecules. They're massive molecules. They're huge proteins, lots and lots of atoms in them. But how do we know what the structure of them is? Well, actually, for a long time, the way we did it was, if you click on there, David, it was actually using something called X-ray diffraction. We had to come back to this diffraction technique again, where a bit like this, where if you've got some sample, such as this diffraction grating, then it spreads the radiation out. If you do it with X-rays, X-rays also have a very short wavelength, about the same size as atoms. You can actually get diffraction from the atomic arrangement. And this is actually the diffraction pattern from DNA that Rosalind Franklin took, that was then used by Crick and Watson to determine the double helix structure of DNA. So this was the data that was actually used to determine the double helix structure of DNA. The problem is, if you're going to use this diffraction technique to work out the structures of these molecules, you've got to crystallize them. You have to get the molecules somehow to form a regular crystal, otherwise you don't get diffraction. So it's been always very tricky trying to get crystallization to occur, and lots of people spend lots of time in laboratories making crystallization of molecules occur. And it's been a real problem. So let's move on a little bit. What we've been able to do now, though, through something called single particle analysis, is actually form electron microscope images of biological structures like this. This is again the coronavirus, and you can see the spike proteins, and it's this sort of data that's led to the sort of structures that you see in the media and so on. But there's a real issue. There's a real problem actually with looking at biological materials. As I mentioned earlier, if you were sitting in an electron microscope, I wouldn't be able to fit you into my electron microscope anyway. The samples are much smaller than you are, so I could squeeze you in. But if I could, you wouldn't last very long under that electron beam. The radiation would be way too high. Same thing for a virus. You put a virus into the electron microscope, and if you just hit it with a number of electrons that I would normally use for one of my, say, gallium nitride experiments, it will just explode and just fry it, basically. It will just damage way too quickly. Also, viruses like to live in biologically relevant solutions, aqueous solutions. If you dry them out, you put them. If you can't put an aqueous, you can't put a liquid into my microscope because the microscope's under vacuum. The liquid immediately just vaporize and contaminate my microscope and make a real mess, and it will dry out, and the virus won't like being dried out. So that doesn't work either. So what do you do about it? You basically want to try and control this damage, and you also want to somehow be able to look at them while they're still in their kind of aqueous environment. So that's been a huge challenge, and it was essentially solved by these gentlemen, Jacques Dubochet, Richard Henderson, and Joachim Frank. Richard Henderson's a British guy at Cambridge. They won the Nobel Prize for Chemistry for solving this problem in 2017. And this is called cryo-electron microscopy. The cryo basically means very, very cold. And what happens is you start off with your virus in some sort of solution, and you put it onto we have a microscope support grid that the virus sits on, and you kind of just make sure you get rid of a bit of excess liquid with a bit of filter paper. And then you drop the sample using a device known as a Vitrobot that drops straight into a little cold beaker of liquid ethane which freezes it incredibly fast, just freezes it instantly. And the ice that forms is vitreous, is what we call it. It means it hasn't had a chance to form ice crystals. It hasn't got a regular array of water. It's randomly, the water molecules in the ice are all randomly oriented. And that means it actually is easier to see the biological macromolecule or the virus that is surrounded by this ice. And also, because it's cold, that actually makes the damage a little bit less as well. The atoms don't tend to get knocked around so easily by the electron beam when it's cold. And we also have now much more sophisticated techniques for controlling the number of electrons that we need to form the images. So we buy these microscopes. This is called a cryo-electron microscope. So you once you plunge this into the liquid ethane, you then transfer it while it's still frozen into this microscope, and then you can look at it in there. These are cryo-electron microscopes. They are about £5 billion each. And they have sold like hotcakes. There are loads of them. People have bought loads of these because it's just so important to be able to see these biological systems. This is called a cryos instrument. And if you just click on, so that's really unlocks. This is how we can actually start to form images of biological things that are non-crystalline. Just to wrap up very quickly, we've also looked at chemotherapy treatments, drugs, and a lot of these chemotherapy drugs actually have platinum in. So back to platinum again, which you can tell is one of my favourite elements. I keep looking at platinum, and so we can see it in my microscope, and we've been able to see that, for example, this drug cisplatin has little crystallites of platinum in. This is the crystal, but this drug here, oxaliplatin, chemotherapy drug, has a much more random arrangement of the platinums in the drug, and we've also been able to image it in cells that have been dosed. If you just click on. And we can actually go up in magnification. This is a cell. We're actually interested in why chemotherapies cause peripheral pain in patients. We're looking at the effects of these chemotherapies on axons, on the nerve cells. This is the nucleus right here. You see this bright blob here. We zoom in on this. You can see some features here. And if we keep going in higher and higher magnification, we can see individual atoms there. So we can see where the platinum's ending up inside the cell. Actually, it was thought that the platinum will went into the nucleus, but we can see that it doesn't actually. It goes other places. I better stop. I think I've run out of my time by quite a long way. But
if we've got some time for questions, I think.
Yes. So, but if you just got to the next slide, Dave, sorry, I'm just going to put my conclusions.
Oh, perfect.
And so, a big challenge in all sciences actually is to make the structure-property relationship. We want to see structures. We want to see biological structures. We want to see structures in chemistry. We want to see structure in material science and in physics. And modern electron microscopy is the eyes of science, if you like. It's how we can see these structures and supports the developments of new materials, chemicals, and medicine. But the most important thing about electron microscopes, using them is really, really good. But seeing atoms and things in a microscope is really, really good. So if you just click on, maybe I'll finish here with my final slide, which is a picture of me having fun and using my electron microscopes. That's taken from a very high angle. Don't know why. Some journalists came to get a picture of me. Climbed up the step ladder, took a slightly weird picture of from height, almost toppling off in the process. So, but that's the electron microscope. So I shall stop there.
Thank you very much for that. I think a round of applause first of all. Are there any questions? I'm just looking on for those who are on Teams. Please feel free if you want to send me any questions, and I'll pass them on. But I think Rachel, who's our vice principal, just going to.
Yeah.
Thank you. I won't be very long. It's
No,
so first of all, it's just been an absolute honour really to be part of this wonderful event, and just a little personal thing really from me, a little known fact. I too am a material scientist.
There we go.
I'm a biomedical material scientist, which is a specific type of material scientist, but I was at University of Birmingham, so there we are. So I spent a lot of hours looking down electron microscopes and really enjoyed that today, so thank you. So on behalf of the organisation, I'd just like to extend my heartfelt thank you for you to be part of this. And thank you for just a wonderful and insightful talk. And I think I'll talk on behalf of everybody here. Massive thanks to David Jones, who's our Northwest STEMnet coordinator and our High Achievers coordinator here at Blackburn College. He's organised tonight's events, including the live streaming, which for those online. So we're streaming to Manchester. Hi everybody, and also Cheshire South and West College, for part of our other Northwest hubs. Can I just ask for a massive round of applause finally for Professor Pete Nellist and also for our people who've organised tonight. So thank you very much.
Thank you, Dave. I don't know quite how, if anybody has touched on you can ask anything actually. You can ask what it's like to be a scientist, you can ask about university life, One of the things that I should have mentioned, Anna Patenik, who's come with me from my college in Oxford, she's our outreach officer. You know, one of the questions she was always asked is, where do you do your laundry? You know, when you're using anything like that, feel free to ask if anyone's got any questions, or if you're being shy, you can always ask me one-to-one afterwards if you'd like. Anybody, anything you'd like to ask? Oh, the question, yeah. Has there been a time where I hated my job? Yeah. Yeah, it's quite a few. I mean, like any job, right? What causes me frustration? There is a lot of administration that comes. You know, you have to spend time writing grant proposals to get funding for your research. You have to write a lot of reports on things. You have to deal with all sorts of weird and wonderful IT systems that the university likes to introduce that I still haven't quite got to grips with. I'm sure it's and so yeah, there's days where you think, oh, but most of the time it's actually a really fun job because it's the one that some days I'll be on the microscope looking at atoms, and then in the afternoon I might be actually with my students doing some teaching. You know, or I might do, you know, it's very occasionally I have days like this where I get out and come do outreach and so on. So it's a very varied job. So most days, you know, it's enjoyable. But there we go, like any job, there's days where it's just some days it's just paperwork. Yeah.
Have you ever been interested in some other field apart from science?
Well, that's or you mean apart from science?
Or has it always been science for you?
It's always been science to me. Yeah. What I would say is that my first degree was actually theoretical physics, actually. Now I'm an experimental material scientist. So one thing I would say to people is that the degree you do doesn't dictate what you're going to do with your career. What you learn in a good science degree is what we call transferable skills. It's about being able to use evidence to make rational deductions and to make predictions about how things might work and develop new things. And those skills you can move between different topics. So, for example, Richard Henderson, who I mentioned is one of the 27 chemistry Nobel Prize winners, his actually first degree was like me, was actually in theoretical physics. He got the Nobel Prize in chemistry, and he actually works in the lab of molecular biology. So, you know, he's somebody who's actually really moves around in science, and that happens a lot actually. That people move around. I mean, for example, the work we're doing with batteries, which I haven't had a chance to talk about, there's a lot of electrochemistry in that. I didn't really know any electrochemistry really until I started research in it, and so then you have to go and learn it, and you use the scientific skills that you've got to actually fairly quickly be able to learn a new scientific area such as electrochemistry. So yeah, people often think that you know, the degree is going to dictate what sort of job you do. A good science degree doesn't do that actually. What a good science degree does give you so many useful skills. You can apply them in all sorts of areas. And my students, when they graduate, some of them go into industry and do research, some stay in academia and do research, some go and become, say, patent lawyers, dealing with development of patents and how you actually protect new developments and so on.
What employability skills do you think that you get from science A levels and science B tech?
Yeah, I mean, it's really about it's analytical skills. It's about being able to look at things and actually be able to understand or look at data and think what you can learn from it. You know, actually how, and you know, for material science, it used to be the case that a lot of our students were going to financial services. That's sort of less so now, but even financial services, actually, they really like to hire scientists because of the skills that they can bring in terms of being able to make deductions, look at data and learn things. So it, you know, it opens up all sorts of different careers having their scientific skills. Yeah. Anybody else?
Did you study at Oxford?
No, I studied at a place further east that's sort of damp, begins with C. We don't like to mention it too often.
Colchester.
Yeah, Cambridge. As in my last talk, when I was with you, your age. I mean, I was just at a regular comprehensive school, and you know, thought I'd do physics. I didn't really do enough research about what topic to do. Actually, I just did physics because I was good at A level, which is not a good reason for doing it. And I should have done more research about subjects. But I was being encouraged to apply for Cambridge, and because I just believed all the myths and it'd become for posh people and all this kind of stuff, I sort of wasn't sure if I wanted to go. And as I did, in the end, I had my arm twisted and I went and loved it, and it was just great fun. And I met a lot of really great people there, had a lot of fun. So I did my first degree at at Cambridge, but as I say, I've worked at various. I've worked at two other, Oxford, at Birmingham, and Trinity College in Dublin. So I've been in quite a few different universities as well as an academic. Yeah.
Should say Marion was just in Oxnet last year.
Oh yes.
She was actually on Peter's strand, weren't you, on the arts and humanities? So she spent some very intense times with Peter Klaus.
We all spent intense times with Peter Klaus. I know Peter very well. Yeah. Good.
Okay. Right. Well, thank you very much, everybody, for coming out this evening. It's been fantastic to get people in here for a public lecture, which is brilliant, and to sort of spark off that the Northwest Science Network in our area. So thank you very much. So I think Hayley, was there anything else you wanted to add, or?
No, I was just going to say thank you very much, David, for organising.
Yeah, thanks, David.
Thank you, Pete, for coming, and Hannah and Chris. Really nice to meet you. And thank you to all the students and staff that have come as well. Well done.
And thanks for staying late. I did waffle on a bit there, but
That's fine. Technical issues at the start.
yes.
Yeah, but thank you very much.